Hello, everybody. Welcome back to another exciting edition of Ed Puzzle Lecture Notes. Today's daily objectives. Number one, when you leave today, you should be able to tell me the difference between autosomes and the sex chromosomes. They're both DNA, they're both chromosomes, but there are differences between the autosomes and the sex chromosomes. Number two, what is a sex-linked trait? Describe what a sex-linked trait is, and be able to give some examples of sex-linked traits. And number three, once again, using Punnett squares, be able to determine the genotypic and phenotypic ratios of sex-linked traits. All right, so we got 16 days left of biology. We got a lot of stuff to do, so let's get some work done, and I hope you enjoy. Sex-linked traits. Sex-linked traits. Remember, traits are any physical characteristic which you can identify: hair color, eye color, height, any of those things. Those are all traits. Now, whether you're a male or female, your first 22 pairs, so chromosome pair number 1 through 22, remember we have 23 pairs, which gives us 46. Well, the first 22 are exactly the same, whether you're a male or a female, boy or a girl. We call those the autosomes. Autosomes, because they're the same whether you're a male or a female. That 23rd pair of chromosomes, those are the sex chromosomes. Those are not the same whether you're a male or female. For those of you that are male, like me, we're XY. We have an X chromosome. I wonder who we got that from. We got that from our mom, because females only have XX. And we got the Y chromosome from our dad. Females, you got one of your X chromosomes from your mom, and your dad happened to give you his X chromosome, which gave you the genotype of XX. Now, if you can look over here, here's the X chromosome that this is a male, because he's got a Y chromosome. Hopefully you can see that the X chromosome is way larger than the little tiny Y chromosome. Now, these chromosomes do more than just determine whether you are male or female. They also carry the genes, anywhere in here, they carry the genes for certain traits. Well, obviously, the X can carry a lot more information than the little tiny Y. All right, so doing a little Punnett square to determine the possible outcomes of if you have a male or a female. Well, that's pretty easy. We're just going to use the sex chromosomes, XX, that'll be the mom, and we're going to cross that with, of course, a male, XY. And just like any Punnett squares, we'll put the females, the moms, on the side. All she has is 2 X chromosomes. We'll put the males on top. He has an X or he has a Y. So just fill in the boxes. XX, that's a girl. XX, another girl. XY, that's a boy. XY, that's a boy. So in case you didn't know, your chances of having a boy, 2 out of 4 are 50%. And your chance of having a girl, also 2 out of 4 or 50%. All right, so like I said, sex-linked traits. These are traits whose genes are carried or found on the sex chromosomes or the 23rd pair. These genes are usually found on the X chromosome because it is much larger than the Y chromosome. The Y chromosome is smaller, so us males, we're missing some genetic information. Females have all the DNA, all the genetic information. But us males, since we have that Y, we're actually missing some DNA. We don't have as much. As a result, sex-linked traits, and these are usually things that aren't that good, they tend to be expressed way more in males than they are in females. And we'll get to that in a second. All right, so 3 traits, sex-linked traits that you don't really need to know what they are, but you kind of do. Color blindness. That is not that you see the world in black and white. You're just unable to distinguish between certain colors like red and green. You kind of see them as the same. There's another trait called hemophilia. That is where if you get a cut or you're bleeding, you have trouble stop bleeding. Your blood won't clot, so you tend to keep bleeding, and that can be serious. And then there's Duchenne's muscular dystrophy. That is like a wasting away or a weakening of all your muscles. It's going to be these two that we're going to talk about most. We're not going to really talk about muscular dystrophy. All right, and here it is again. Color blindness, inability to distinguish colors. You don't really need to write this. I mean, you should kind of know what that is, but you won't need to know that. Hemophilia, it's an inability for blood to clot properly after a cut. So if you start bleeding, it's really hard for you to stop. So if you have kind of a serious cut, that can be a pretty serious deal. And muscular dystrophy, once again, it's a progressive weakening and loss of skeletal muscles. But basically, you're going to have to know how to do the Punnett squares more than you're going to have to really know what these things are. All right, so here's a little unofficial colorblind test. Does not mean you're colorblind if you can't see it, but if people that are colorblind, when they look at this figure over here, they cannot see that there is an 8 in here. Now, once again, it does not look like it's in black and white. They just can't distinguish. They can't tell the difference between these colors. Same with this one over here. They would not see the number 56. It would simply look like a colorful circle. They can't see it. And this one on the bottom is the hardest one to see, but there's a 5 in there. So that's how they tell if you're colorblind or not. They give you shapes like this, and they see if you can determine the number or the letter or whatever that's inside of it. All right, so here is what our new alleles are going to look like. Remember with blood types, we had the big I and A and like big I B. Well, we no longer have the big I for isoagglutinogen. Now we need the XX and we need the XY to see if it's a male or female. So the first one we're going to do is color blindness, red-green color blindness. You have trouble distinguishing the difference between red and green. They kind of look the same. So over here, we have a totally normal female. You can see she's a female because she's got XX, and the capital N is normal vision. Being colorblind is recessive, so you got to have 2 little n's in order to be colorblind. So this female is totally normal vision. The next female down, you can see the XX, so she's a female. She has 1 normal allele and 1 colorblind allele. But since normal vision is dominant, she has totally normal vision, but she is a carrier of the colorblindness. So she can pass that on to her kids. The only way for a female to be colorblind is this one right here. She's XX again, so that makes her a female. And notice she has 2 little n's, 2 recessive alleles. So that is the only way a female can be colorblind. She has to get the recessive colorblind allele from both of her parents. On the other hand, with us males, we only have 1 allele for color blindness. It's on our X chromosome. Remember, the little y, it's too small. It's not big enough to carry that gene for color blindness. So for a normal male, you get the normal vision allele from your mom, and your dad gives you the Y, but it doesn't even have that information. So there is no N when it comes to the Y. For a male to be colorblind, it's all determined by the mom. If the mom gives that colorblind allele, the dad gives the Y, which does not even have an N with it. So the males, it's basically you get 1 shot. If the mom gives you a colorblind allele, you're colorblind. Where females over here, the mom could give a colorblind allele, but if the dad saves the day and gives the normal allele, she won't be colorblind. So that's why sex-linked traits are way more common in males than in females. All right, so let's try a couple of these. The first one, we're going to do color blindness. We're going to cross a female who happens to be colorblind with a male who has normal vision. Let's start with a female. First, to show she's a female, we're going to start with XX. She's colorblind, so little n, little n. She's got to have 2 recessive alleles in order for her to be colorblind. And we're going to cross her with a normal vision male. So to make him a male, we're going to put X, Y, and he's normal, so he's going to have one big N. Now, one big N. Do not give me another allele right there. That's the first thing, one of the big mistakes people make. Do not give me another letter by the Y. The Y chromosome does not hold this gene or this trait, this allele for color blindness. All right, so here we go, just like any Punnett square. We'll put the female up top, X little n, X little n, and we'll put the male on the side, X, he's got a capital N, normal allele, and he's got a Y. All right, fill in the boxes. X big N, X little n. This is going to be X big N, X little n. Down here we're going to have X little n, Y, nothing. Remember, nothing on the Y. And this one's going to be X little n, Y, nothing. All right, so what are these kids going to be? Once again, here it is. The females are both going to be X big N, X little n. But since they have the dominant allele, 100% of girls are going to be normal. Now they're going to be carriers. They do carry that recessive colorblind allele. They can pass that on to their kids, but themselves, they're not colorblind. The males, on the other hand, 100% of the boys they have, all of them are going to get that recessive colorblind allele from the mom. So 100% of the males will be colorblind. Now we'll try one on the bottom. This is going to be for hemophilia. Now we're going to use H. Big H is normal, dominant, normal. You're not a hemophiliac. And the recessive little h is the allele for having hemophilia. We're going to cross a female that is not hemophiliac, but she is a carrier. She does carry the recessive bleeder, quote unquote, allele. And we're going to cross that with a totally normal male. All right, so let's start with a female. She's not hemophiliac, so we're going to start with XX. She is not hemophiliac. She has a capital H, but she does carry the recessive hemophiliac allele. We're going to cross her with a totally normal male. So to make a male, we're going to start with X, Y, and he's normal. He's going to have a big H. Now remember, do not give me anything on the Y. The Y is too small. Do not give me any allele on the Y. All right, then it just becomes a normal Punnett square. Let's put the female on the top. X, big H, X, little h, recessive. Male on the side. X, big H, and his Y. Filling it in. X, X. These are going to be both big H's. This female is going to be X, big H from dad, X, little h from mom. This male down here, X, big H from mom, and he obviously gets the Y from his dad, which does not carry the allele for hemophilia. And this male over here, X, little h, and Y. Now let's see. Once again, 100% of the females will not have hemophilia, even though this girl over here got the bleeder or hemophilia allele from the mom. The dad saved the day because the dad, all he has is a normal big H allele. Now for the boys, it's a little different. This boy over here, he was lucky. He got the normal allele from his mom. The mom could have gave either one. And this guy lucked out. This guy over here in the corner, on the other hand, did not luck out. He received the hemophilia allele. So, when it comes to males, 50% will have hemophilia and 50% will be totally normal. All right, now a little chromosome mapping. Genome mapping. So, they just recently, well, not recently, it was like in 2001, they actually sequenced the entire DNA sequence in the human genome It's called the Human Genome Project. But we're going to do a little something that's not quite as complicated. But there is ways, like scientists now, they know where most of these genes are located. You know, they could say, hey, that's where dimples is at. They can say, hey, that's where a thing called fish odor syndrome. Yes, that is a real thing. If you have the gene or allele for fish odor, you actually smell like rotten fish. I think it's on chromosome number 1, but I'm not sure. But they know where these genes are at. They've located them. Now I'm going to do a little simpler method, and we'll see. This is going to be kind of confusing, so don't get too freaked out. We're going to try to work through it on a worksheet. And if you don't understand it, don't worry too much, because it will not crush your grade. The Punnett squares. Punnett squares. Remember, that is where it's at. Make sure you know how to do those. Alright, so I don't even know if I should explain this, because it's super hard and complicated, but we're going to try our best. Alright, first we got to remember crossing over. Remember, that is where homologous chromosomes that you can see here during meiosis, remember in meiosis, where they actually exchange a little DNA. The homologous chromosomes, you can see the mom and dad chromosomes actually exchanged a little bit of their DNA. Well, the A, B, and the C and all those represent different genes. Like the A is way up here, the B is here, and the C is there. Well, geneticists can actually map genes by how often these letters or these alleles get separated from each other during meiosis. For example, this is a deck of cards, right? Let's say you just took 2 cards out of the deck. You took the Ace of Hearts and the Ace of Clubs. And on the first time, you placed them back in the deck, and you put the Ace of Hearts way up on the top, and you put the Ace of Clubs way down on the bottom. Then I asked you to cut the cards, which means to pick up some of the cards and separate them into 2 stacks. If the Ace of Hearts and the Ace of Clubs are really far away, far apart from each other, you're going to cut them away from each other almost 100% of the time. The only way you wouldn't is if you cut it way down at the bottom and you happen to get it just above where the Ace of Clubs is. On the other hand, if you put the Ace of Hearts and the Ace of Clubs right next to each other, in that deck of cards, right next to each other, so like one's right there and the other one's just right below it. Now I ask you to cut the cards. You may cut it 100 times in a row, but you may cut it a little too high or a little too low, and the Ace of Hearts and the Ace of Clubs stay together. It would take the perfect cut to actually separate those 2 from each other. Well, it's the same thing that happens in chromosomes. The further these alleles or these genes are away from each other, the more often they will get cut, separated during crossing over. The closer they are, like say the B and the C down here, the closer they are, if you cut it up here, the B and C stay together. If you cut it right there, the B and C stay together. Now, both of those times, the A and the C and the A and the B would get separated, but the only way you'll separate the B and the C is it's got to be right there, like that shows right here. So it's much harder to cut the chromosome during crossing over in between the B and the C than it is between the A and the C. And the more often they get separated from each other during meiosis, the farther away they are on the chromosome. Now, if they get separated from each other exactly 50% of the time, that tells geneticists that they are not on the same chromosome. That the only reason they're getting separated exactly 50% of the time because of independent assortment. Remember, independent assortment is like a coin flip. Which chromosome you're going to give, and a coin flip is 50%. All right, now we're going to try one, and it's really not that hard. It's kind of like solving a puzzle. So you can see here is a chromosome right there, and you can see these are the different alleles or genes. You got C, P, Q, and D. Now these little black stripes on that pink chromosome. Those are where the genes are located. The question is, where's C, where's P, where's Q, and where's D? Here's the trick. You always start out with the ones that are the furthest apart. D and C get separated from each other 33% of the time. So since they get cut away from each other the most often, 33% is the biggest number there, we know that they are the farthest away from each other because they get separated from each other the most. So we're going to put D out here and we're going to put C right there. Those are the two farthest genes away from each other. Now I'm going to go to this one right here. P and Q get separated from each other during meiosis from crossing over 18% of the time. So that's going to be the second largest distance. I can see that this right here is the second largest distance. C and D is the farthest apart, but that's the second furthest genes away from each other. Question is, where does P go? Is P right here or is P right there? Well, you can see from this one right here, P is very close to C because it only gets separated from P 8% of the time. Therefore, I know P has to be there. That only leaves Q, and Q would be right there. All right, I'll try this one right here. Remember, key is start with the furthest away from each other. Y and P are 45, and these turn into map units called centimorgans. Well, you have to know that? No, but I don't know. Some people like to know. Centimorgans. So Y and P are 45. They get separated from each other 45% of the time. That's huge. You know they're way far away. So we're going to put Y way out here, and we'll put P way out here. Remember, it cannot be over 50% because 50% means they're not even on the same chromosome. Now, I'm going to see where D's at. D is 40. D is really far away from Y. So D is going to be somewhere real far away from Y. We know it's not this way because it can't, that's too far. 40, it's just going to go off the chromosome. So we know it's going to be this way. So 40 is almost going to be as far as P is away over there. So we'll put D right about there. Now, the only one we got left, where is R? Once again, R gets separated from D 25% of the time. That's pretty far. They got to be pretty far away to get separated from each other that often. So we know we're not going to go this way to find R. We got to go this way. And 25% would be somewhere around in there. So that would be where R is. Once again, just understanding the concept is kind of the most important thing. Understanding that geneticists can locate where genes are at and if they're on the same chromosome by looking how often they get separated from each other during meiosis. The further away they are from each other, the more often they'll be separated because it is hard for crossing over to happen exactly right there between D and P. But it is really easy to separate between P and Y. Hopefully this made a little bit of sense to you. If not, don't worry about it because the gene mapping is not going to be that big of a deal on our exam. Thank you very much for listening, and I'll talk to you next time.